Hello, I'm Professor Sims, and in this video I will discuss evolution of populations. This is the 10th in a series of 10 lessons held as part of my general genetics course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include defining population genetics, its correlation to population evolution, talking about the Hardy-Weinburg principle and its importance. We'll describe the different types of variation in a population. We'll explain how natural selection determines heritable variation. We'll discuss genetic drift and the bottleneck and founder effects. We'll explain how evolutionary forces influence a population's allele frequency, and we'll explain the different ways natural selection can shape populations. All life on Earth shares a common ancestry according to the theory of evolution. Humans, beetles, plants, and bacteria have evolved over millions of years from a common origin into the diverse organisms we see today. Evolution is a key concept in understanding life. It's driven by natural selection, which favors advantageous traits for survival and reproduction. While natural selection cannot create new traits, other sources of variation, like mutations, contribute to the changes in population and species. Over time, the integration of genetics and evolution led to the modern synthesis, explaining how natural selection affects genetic makeup and leads to gradual changes in populations and the emergence of new species, which is called macroevolution. Population genetics is a branch of biology that studies the genetic composition and variation within populations of organisms. It focuses on understanding how genes and alleles are distributed and how they change over time within a population. Population genetics aims to explain the patterns and mechanisms of genetic changes in populations and how these changes can lead to evolution. Population genetics plays a significant role in understanding natural selection, the driving force behind evolution. Natural selection favors advantageous alleles that enhance an organism's fitness for survival and reproduction, leading to an increase in their allele frequency in the population. Population genetics also investigates genetic drift, which is a random process that can cause allele frequencies to change, especially in small populations. Genetic drift can lead to random fluctuations in allele frequencies and is more pronounced in smaller populations. Over time, genetic drift can result in evolutionary changes. As can gene flow. Gene flow is the movement of alleles between populations through migration and interbreeding. Gene flow can introduce new genetic variation into a population or counteract the effects of genetic drift, thereby influencing the evolutionary trajectory of populations. Mutations are the ultimate source of genetic diversity, and they contribute to the pool of potential alleles available for natural selection to act upon. Finally, population genetics studies how population structure and geographic barriers can affect gene flow and lead to the emergence of new species. The Hardy-Weinburg principle, also known as the Hardy-Weinburg equilibrium or the Hardy-Weinburg law, is a fundamental concept in population genetics. It describes the relationship between the allele and genotype frequencies in a population under certain idealized conditions. The principle provides a null model against which real-world populations can be compared in order to detect evolutionary changes and understand factors influencing genetic variation. Essentially, if a population is in Hardy-Weinburg principle, what that means is that nothing is changing. Natural selection is not acting upon that population. It's in fact in equilibrium as far as its population genetics are concerned. The Hardy-Weinburg principle is based on several key assumptions. First is large population size. The population has to be large enough that random sampling errors do not significantly affect allele frequencies. The next one is no mutation. There shouldn't be any new mutations introducing new alleles into the population. No migration, no immigration or immigration is influencing the gene pool. Random mating, so it's assuming that mating within the population occurs randomly with respect to genotype, meaning individuals do not preferentially choose mates based on their genotype. And then there's an assumption of no natural selection So no advantage or disadvantage is associated with specific genotypes in terms of survival and reproductive success. So if a population is in Hardy-Weinburg principle, all of these things are true if they are in equilibrium. So under these assumptions, the Hardy-Weinburg principle states that the allele and genotype frequencies within a population will remain constant from generation to generation. It describes the mathematical relationship between the frequencies of two alleles, let's call them A, big A, little a, and the frequency of three possible genotypes, big A, big A, big A, and little a, in a population is p squared plus 2pq plus q squared equals 1. Also, p plus q equals 1. So p represents the frequency of the big A allele, q is the frequency of the little a allele, p squared is the frequency of the big A genotype, and q squared is the frequency of the little a genotype. Let's watch this example of how to calculate allele frequencies and genotype frequencies using the Hardy-Weinburg equations.
All frogs in this population will be either big G, big G little g, or little g. There's also an allele frequency in our population. We're going to say here that 60%, a frequency of 0.6 of the alleles are big G. 40%, or a frequency of 0.4 of the alleles are little g. Notice the percentages add up to 100%, and the frequencies add up to 1. Now, to be in Hardy-Weinburg equilibrium, we have to have 5 assumptions. Number 1, no selection. No natural selection is acting upon these frogs. That means neither dark green nor light green will have any impact on reproductive fitness. Number 2, no mutation. Baby frogs inherit genes from their parents and there are never mutations. Number 3, no migration. Frogs can't come in, frogs can't go out. Number 4, large population. There are a lot of frogs. Number 5, random mating. The frogs mate without any specific choice. All of these 5 assumptions must be kept in order for Hardy-Weinburg equilibrium to happen. So, in real life, does this generally happen? No. For example, in real life, maybe in a certain environment, predators can more easily see the light green frogs, so perhaps they are eaten more and have less reproductive fitness. So if Hardy-Weinburg equilibrium is unrealistic in nature, why does the Hardy-Weinburg equilibrium matter? That's where the math comes in. The Hardy-Weinburg equilibrium gives you this baseline to compare how an evolving population could compare to one that remains constant without evolutionary forces acting upon it. And now let's explore the math. So there are 2 equations in Hardy-Weinburg equilibrium that we will focus on. We're going to start with the first one. P plus Q equals 1. In this equation, P is the dominant allele frequency in the population, and Q is the recessive allele frequency in the population. By the way, Hardy-Weinburg equilibrium doesn't mean that P has to equal Q, and the dominant allele frequency in any population doesn't have to be larger than the recessive allele frequency in a population. That's a misconception because dominant alleles aren't always the more common allele. The equation does say that the dominant allele frequency and the recessive allele frequency have to equal 1. So in my example here, if I say 60% of the alleles are big G, 40% of the alleles are little g, 0.6 is P, and 0.4 is Q. So this equation, P plus Q equals 1, is for those allele frequencies. But what if I wanted to know the genotype frequencies? Meaning, I wanted to know the frequency of frogs that are homozygous dominant, which is big G, heterozygous, which is big G, or homozygous recessive, which is little g. Then I can use this other Hardy-Weinburg equilibrium equation, P squared plus 2PQ plus Q squared is equal to 1. So P squared is the homozygous dominant frequency, the frequency of big G in this case. 2PQ is the heterozygous frequency, so big G frequency in this case. Q squared is the homozygous recessive frequency, so little g frequency in this case. Let's plug those previous P and Q values in. P squared would then be 0.36. 2PQ would be 0.48. Q squared equals 0.16. Cool, huh? The only thing is that when calculating Hardy-Weinburg equilibrium problems, you don't always have the P and Q values. So we're going to do an example. New population of frogs, so forget the previous frequencies. This is a new frog population in a new frog land, but we'll still use the same allele letters. So here's the information you get for this new population. There are 500 frogs, and of those, 375 frogs are dark green. The rest are light green. With that information, please solve all genotype frequencies and all allele frequencies if in Hardy-Weinburg equilibrium. Step 1. Determine first whether you're going to work with the first equation or the second equation. Since I'm working with individuals here that have genotypes, I'm going to work with the second equation first. Step 2. Figure out what value you can determine. So 375 frogs are dark green out of 500 total. That would mean 125 frogs are light green since there are 500 frogs total. But I can't use whole numbers like that, as both of these equations ultimately are equal to 1. I need frequencies for these equations. Now, I could say 375 dark green frogs out of 500 total frogs is 0.75, but all I know is that would be the frequency of dark green frogs. The problem is dark green frogs could have genotype big G, or they could have big G, little g. I can't assume they're one genotype or the other, so I shouldn't use that value. The recessive genotype, resulting in a light green trait, is safe to use though, because I know that light green frogs are the genotype little g. They can't be anything else. So 125 frogs out of 500 frogs equals 0.25. That means, from the equation, the value q squared equals 0.25. Step 3. Take a value you solved from the previous step and calculate from there. So if I know that q squared is 0.25, then I could go ahead and solve for q. If q squared is 0.25, I can determine q if I take the square root of 0.25. Therefore, q is equal to 0.5. That's the allele frequency for the recessive allele little g. If I know the q value, I can find out the p value using the first equation. Since p plus q is equal to 1, now I know that p is equal to 0.5. That's the allele frequency for the dominant allele, big G. Now I have P, and I have Q, and I can take care of everything else. I can use the second equation to determine the homozygous dominant frequency, the heterozygous frequency, and I already knew the homozygous recessive frequency from the beginning. Plugging those in, P squared equals 0.25 for the genotype frequency big G, 2PQ equals 0.5 for the genotype frequency big G, Q squared equals 0.25 for the genotype frequency little g.
By studying allele and genotype frequencies, population genetics helps us understand how genetic variations contribute to the diversity of populations and how evolution shapes these populations over time. In theory, if a population is in equilibrium with no evolutionary forces at play, its gene pool and genetic structure would remain constant from generation to generation. In reality, though, natural populations are constantly evolving due to various factors like drift, mutation, migration, and selection. While the exact distribution of phenotypes can only be determined through direct observation, the Hardy-Weinburg principle provides a mathematical reference point of a non-evolving population against which evolving populations can be compared. Deviations from the expected frequencies of alleles or genotypes indicate evolutionary changes. That figure 19.2 is showing that when populations are in Hardy-Weinburg equilibrium, the allelic frequency is stable from generation to generation, and we can determine the allele distribution from the Hardy-Weinburg equations. If the allelic frequency measured in the field differs from the predicted value, then scientists can make inferences about what evolutionary forces are at play. Populations often exhibit phenotypic variations known as polymorphisms, where individuals express different alleles of a particular gene. These variations are influenced by factors like genetic structure and the environment. Understanding the sources of phenotypic variation in a population is crucial for predicting its evolutionary response to different pressures. Natural selection and other evolutionary forces can only act on heritable traits, those that are encoded in an organism's genetic makeup. Alleles that can confer advantageous traits may be selected, while non-heritable acquired traits that do not contribute to evolution would not be selected. For example, a parent's workout routine and increased muscle strength won't necessarily pass on to their offspring, but genetic factors related to traits like speed could be inherited. Figure 19.3 is showing an example of the distribution of phenotypes in a litter of kittens. So I didn't really have anything else to say about this. I just wanted to show you some cute kittens. Heritability refers to the portion of variation in phenotypes that can be attributed to genetic differences among individuals in a population. When a population exhibits high heritability, it means that genetic variation strongly influences the traits and makes them more susceptible to evolutionary forces. Genetic variance, which encompasses the diversity of alleles and genotypes within a population, is important when it comes to breeding programs for species preservation. In these programs, scientists aim to maintain high genetic variance to preserve the diversity of traits. This approach also helps minimize the risks associated with inbreeding, where closely related individuals mate and bring together harmful recessive mutations that can lead to abnormalities and disease susceptibility. For instance, rare recessive alleles causing diseases may persist in a population at low levels, since the chance of carriers mating and producing affected offspring are low. However, when carriers interbreed, the likelihood of disease inheritance increases, resulting in a phenomenon known as inbreeding depression.
Inbreeding limits the available gene pool and can increase the chances that you'll pass on harmful recessive traits to your kids. And just to clarify, a recessive trait is one that's only expressed if you inherit it from both your parents. You see, in humans and lots of other organisms, we get one set of genes from our mom and one set from our dad. So if your dad has a harmful recessive version of a gene and passes it on to you, you won't express it unless your mom gives you a copy of it too. Consequently, these genes can hide for generations, secretly being passed down but never expressed. But if your parents are closely related, the chances that they both carry the harmful version of the gene are way higher. And that's how you end up with people like the famously inbred Charles II of Spain, who was allegedly sickly, infertile, and had a tongue so big that he drooled and could barely speak. If inbreeding lowers the fitness of a population, scientists say the population is experiencing inbreeding depression. That being said, mating with partners who aren't genetically similar to you is risky too. If your traits make you really well suited to your environment, why mess with a good thing by mating with someone whose genes might be super different? If instead you mate with your relative whose genes are similar to yours, you may be ensuring that those good traits get passed on. And in nature, when the costs of mating with unrelated partners outweigh the fitness benefits, inbreeding may be a better option. So what's going on with the fig wasps? Well, let me first be clear that there are hundreds of different species of these guys, and many of them don't exclusively inbreed. But I spoke with a scientist, and here's what I can tell you in a general way about why inbreeding might be a good strategy for some of them, though there may be other factors at play. Okay, so fig wasps are what are called haplodiploid, which means that their offspring don't necessarily get a set of genes from each parent. The females do. They get a set from both their mom and their dad, but the males only get one set of genes, and it comes from their mother. This is actually true of most members of Hymenoptera, which is the order of insects that includes wasps, bees, and ants. Because males only have one set of genes, recessive traits really can't hide for long in a population. Males will outwardly express whatever traits their mom gives them. And if those traits make them unhealthy, then it's less likely they'll be able to mate and pass their genes on to the next generation. In that way, undesirable genes are more easily weeded out of the population in a haplodiploid species, making inbreeding a safer practice, and one that can preserve the good genes as well. Now, inbreeding is actually not as rare in nature as you'd expect. Some species do it simply because it's really hard to find a mate, and lots of plants self-fertilize, which is basically the ultimate form of inbreeding. All that said, genetic diversity is a wonderful thing. It can make populations more resilient in the face of change or hardship. But expanding your gene pool is not always the best strategy for survival. As a wise man once said, life, finds a way. And for fig wasps, that way is sometimes inbreeding.
Inbreeding depression occurs because of a quirk of natural selection and genetics. As natural selection acts on a population, it weeds out genes that have advantageous effects. But it can only weed out these genes if they're actually expressed in an individual. For dominant gene versions, that's no problem. Individuals carrying dominant genes with a detrimental effect will be selected against, and eventually these genes will be purged from the population. For recessive gene versions, however, the story's a bit different. Recessive genes are only expressed when an individual carries 2 copies of them. Once natural selection has removed most of the detrimental recessive genes from a population, these seldomly wind up paired with an identical copy and are effectively hidden from the effects of natural selection. This means that most populations carry many deleterious recessive gene versions that are very rarely expressed, except in cases of inbreeding. Closely related individuals are likely to carry the same deleterious recessive gene versions and pass 2 copies of that gene onto their offspring. Genetic drift, influenced by chance events, causes fluctuation in allele and genotype frequencies within a population. Certain individuals may have more offspring due to being at the right place and time, leading to an increase in frequency for certain genes. Factors like camouflage or drought resistance exert selective pressures as well. Gene flow, the movement of alleles through migration, plays a significant role in evolution because populations experience fluctuations as individuals disperse and introduce new genetic variation in different locations and habitats. Mutations drive population diversity. Harmful mutations are eliminated by natural selection, while beneficial mutations spread. Some mutations have significant effects on genes and phenotypes, while others persist naturally. Natural disasters amplify genetic drift's impact through the bottleneck effect, wiping out a portion of the gene pool. The surviving genetic makeup defines the entire population, causing differences from pre-disaster populations. The founder effect occurs when a subset establishes a new population, aligning the genetic composition with the founding individuals. The Afrikaner population in South Africa is a good example of the founder effect. The genetic history of Afrikaners shows a high prevalence of certain mutations compared to other populations, likely due to a higher occurrence of these mutations among the founding colonists. Consequently, the population has a higher incidence of genetic disorders, such as Huntington's disease and Fanconi anemia, which can lead to various health complications, including blood marrow abnormalities and cancer. Figure 19.4 shows how genetic drift in a population can lead to eliminating an allele from a population by chance. In this example, rabbits with brown coat color, allele big B, are dominant over rabbits with white coat color, allele little b. In the first generation, the 2 alleles occur with equal frequency in the population, resulting in a P and Q value of 0.5 each. Only half the individuals reproduce, resulting in a second generation with P and Q values of 0.7 and 0.3 respectively. Only 2 individuals in the second generation reproduce, and by chance these individuals are homozygous dominant for brown coat color. As a result, in the third generation the recessive little b allele is lost. Small populations are more vulnerable to genetic drift, while large populations are more resilient to chance events. For instance, if an individual out of a group of 10 dies before producing any offspring, their genes, which make up 1/10 of the gene pool, will be lost. However, in a population of 100, the loss would only amount to 1% of the overall gene pool, making it less significant in terms of genetic structure. Figure 19.6 illustrates how gene flow can occur when an individual travels from one geographic location to the other. You can imagine that this individual bug, or a small group of bugs, move from one location to another and they bring their genes with them, which increases the level of genetic variation in the new location. The bottleneck effect happens when the population number is dramatically decreased as a result of natural disasters such as earthquakes, floods, and other types of natural disasters. The new population is made up of the organisms that were able to survive the disaster, and the new population's allele frequency will be radically different from the original allele frequency. Let me show you this very short video that's a very good demonstration of how the bottleneck effect works.
What are you doing, crafts?
No, I'm actually working on a demonstration for random selection. I have selected some beads to represent people, and I have 3 different colors, red, white, and blue, representing a single trait, but I have 3 different versions, the red, white, and blue versions.
Okay, but why did you put them in the bottle?
Well, the bottle is representative of the whole population of people living in a region, for example. We have roughly equal frequencies of all the colors in our population, though there's a slight increase in the number of red individuals living at the sort of top of the bottle. Okay. Now, for this to be representative of random selection, we need an event to happen that's completely random.
What a meteor?
A meteor, that sounds great. So a meteor strike is coming to Earth, and it comes to this region and... Now notice what happened is all these people, a couple still trying to survive there, everybody that survived ran away and got away from the meteor, and those that weren't able to run fast enough stayed in the bottle, and unfortunately they didn't survive. And so the people who survived were able to run away here into the glass dish, and those that couldn't are still here in the bottle. Unfortunately, these didn't survive. And what I want you to notice, if we see here, is that tell me the frequency difference between the individuals in the bottle and those in the glass dish.
Well, it looks like most of the red survived, but not as many blue and white beads did.
Exactly, and that's because there was a change in the allelic frequency due to a random selection event.
Oh, the bottleneck effect.
Exactly. You can see here we've represented that quite literally. And so those in the bottle didn't survive. We changed the allelic frequency, more red in the dish, those that survived. And now we have a change that resulted from a completely random event.
So as we know, the founder effect occurs when a small group of individuals from a larger population establishes a new population in a different location. When a physical barrier isolates a subset of the population, the genetic composition of the new population reflects that of the founding members, the founding individuals, which may differ from the original population. This can lead to higher frequencies of certain alleles and genetic traits in the new population, including rare or harmful mutations. As a result, the new population, including these rare or harmful mutations, may experience higher incidence of certain genetic disorders or unique genetic characteristics compared to the original population. Non-random mating also plays a role in population genetics. So mate choice, such as female peahens preferring peacocks with larger, more vibrant tails, can lead to selective breeding. Assortative mating, where individuals prefer to mate with partners who resemble them phenotypically, is another form of non-random mating. Physical location can also influence mating patterns, particularly in large populations scattered across vast areas where individuals may have limited access to each other due to geographic barriers or distance. The environment is another factor that contributes to population variation. Environmental conditions can impact phenotypes such as darker skin in individuals exposed to sunlight regularly. In some species, the environment even determines major characteristics like sex, as seen in turtles and reptiles, with temperature-dependent sex determination. Geographic separation between populations can result in phenotypic differences observed in variations along ecological gradients known as clines. A cline is a gradual, continuous variation in characteristic or trait of a population across a geographic range or environmental gradient. Clines typically result from the influence of environmental factors on the expression of genes within a population. As individuals move across different geographic regions or encounter varying environmental conditions, their traits may change gradually and predictably. Clines are often observed in response to factors such as temperature, altitude, or other ecological gradients, and they provide valuable insights into how populations adapt and evolve in different environments. For example, warm-blooded animals tend to have larger bodies in cooler climates closer to the poles, enabling better heat conservation, while flowering plants, on the other hand, bloom at different times along mountain slopes. Figure 19.7 is showing a baby alligator, and it's talking about how the temperature at which the eggs are incubated determine the American alligator's sex. Eggs incubated at 30 degrees Celsius produce females, and eggs incubated at 33 degrees Celsius produce males. Natural selection shapes a population's traits by favoring beneficial alleles that enhance survival and reproduction. So while discouraging harmful alleles. And this process is known as adaptive evolution, and it acts on entire organisms rather than individual alleles. For instance, it may possess a genotype that promotes high reproductive ability, but also carries alleles that carry fatal childhood disease. In such cases, natural selection operates at the individual level, favoring these individuals that contribute more to the gene pool of the next generation. This concept of an organism's evolutionary fitness quantifies its reproductive success relative to other members of the population. Scientists employ various approaches to study how selection influences population variation, including stabilizing selection, directional selection, diversifying selection, frequency-dependent selection, and sexual selection. Each of these modes of selection can impact the genetic diversity within a population, making individuals more or less similar and influencing the range of phenotypes observed. Stabilizing selection occurs when natural selection favors an average phenotype and is against extreme variations. For instance, in a population of forest-dwelling mice, individuals with fur color closely matching the forest floor have a higher chance of survival as they blend in better and they're less likely to be detected by predators. Mice with lighter or darker fur will stand out and become more vulnerable to predation. Over time, the stabilizing selection will reduce the genetic variance within the population. Directional selection occurs when environmental changes favor phenotypes at one end of the existing variation spectrum. An iconic example of this is peppered moths during the Industrial Revolution. Originally, light-colored moths blended in with the trees better, but as pollution darkened the trees, birds preyed more easily on the lighter moths, and then the darker moths increased due to their superior camouflage. If the forest floor experienced a color change, a hypothetical population of mice might evolve different colors to adapt to that as well. In such cases, directional selection is shifting the genetic variance of the population towards the new advantageous phenotype. Sometimes natural selection favors multiple distinct phenotypes, while intermediate ones are less advantageous on average. Scientists call this diversifying selection. An example of diversifying selection can be seen in animal populations with different male forms. Dominant alpha males use their strength to secure mates, while smaller males employ sneaky tactics to mate with females. In an alpha male's territory, both alpha and sneaking males are selected for, while medium-sized males who can't outcompete are too big for sneaking, and they face selection against them. Diversifying selection can also occur when environmental change favors individuals on opposite ends of the phenotypic spectrum. Imagine a mouse population living at a beach with light-colored sand and patches of tall grass. Light-colored mice will blend in with the sand, dark-colored mice will be able to hide in the grass, while medium-colored mice wouldn't blend very well with the sand or the grass. This type of selection increases genetic variance and promotes population diversity. Another type of selection, called frequency-dependent selection, favors phenotypes that are either common or rare. So an interesting example of frequency-dependent selection is observed in Pacific Northwest lizards. Male common side-botched lizards exhibit 3 throat-colored patterns: orange, blue, and yellow. Each pattern corresponds to a different reproductive strategy. The orange males are strong and they fight for females. Blue males form strong pair bonds, and yellow males resemble females in order to sneak copulations. Like a game of rock, paper, scissors, orange beats blue, blue beats yellow, and yellow beats orange in the competition for mates. Thus, selection favors orange males when blue dominates the population, blue males when yellow dominates, and yellow males when orange dominates. This cycle of selection results in varying distributions of these phenotypes over generations. Sexual dimorphisms, where males and females of a species differ beyond just their reproductive organs, are common in many populations. Males often possess larger sizes and elaborate adornments, while females often are smaller and less decorated. These differences arise when there is greater variation in male reproductive success. Some males, typically bigger and more adorned, secure the majority of matings, while others are unsuccessful. And this can be due to males' fighting ability or females' preference for certain traits. And such variation generates strong selection pressure among males, leading to the evolution of larger bodied sizes and elaborate ornaments to attract female attention. Females with a smaller number of chosen matings have a greater tendency to select desirable males. Sexual dimorphism varies among species, and some species exhibit reversed sex roles where the females have have greater reproductive success variance and are selected for larger sizes and elaborate traits typically associated with males. Figure 19.8 shows how different types of natural selection can impact the distribution of phenotypes within a population. In stabilizing selection, A, an average phenotype is favored. In directional selection, B, a change in the environment shifts the spectrum of observed phenotypes. Then in C, diversifying selection, two or more extreme phenotypes are selected for, while the average phenotype is selected against. These charts also help to illustrate the fact that natural selection acts on individuals to affect change in a population. Individuals do not evolve, populations evolve. This figure shows a hypothetical example of frequency-dependent selection. So the color of the cat indicates both their genotype and their food source. Black cats eat red apples, green cats eat green apples. I know it's kind of silly, but stick with me. To start with, the incredibly low frequency of green cats means that one green cat can exploit a huge food source compared to black cats. Because of this, natural selection favors green cats. However, in the next generation, evolution overcompensates and produces way too many green cats, and now black cats are getting much more food. Natural selection bounces back to favor the black cats. Eventually, this causes an equilibrium balance of the two cat types, and these changes are reflected in the overall frequency of the two types over time, which eventually evens out. And the bottom right figure demonstrates that for both cat types, the frequency of that color is inversely proportional to the overall fitness, measured as a proxy by amount of food per cat. Here is a clip that helps to better illustrate these different types of selection.
Now, onto the types of natural selection. There are three categories of natural selection: directional selection, where one extreme is favored; stabilizing selection, where an intermediate is favored; and diversifying selection, where both extremes are favored simultaneously. These can be a bit difficult to envision, so we're now going to go over an example and a graph for each one. You might see graphs like this on various standardized tests. They're very popular on exams like the AP, the IB, the A-level, etc. They can sometimes have different colors or orientations, but the most important thing is to remember that one line usually shows what a population used to look like, and the other line shows what the population looked like later on. In this video, the black line shows the starting population, and the green line shows what it comes to look like after being put under pressure from certain conditions. In directional selection, one extreme phenotype is favored rather than an intermediate. Directional selection can be a subtle shift, or it can occur more dramatically, as in the famous case of the peppered moths. Prior to the Industrial Revolution, a population of light-colored moths inhabited the city of Manchester in the UK. These moths blended in beautifully against the neutral-colored tree bark and buildings. Darker-colored moths were virtually unknown, as they were so easy for predators to see. However, by the year 1848, the moth population had almost entirely changed color. Dark-colored moths were now the norm, and light-colored moths were more rare. Why? The city of Manchester rapidly became industrialized right around that time. The new coal-burning factories covered the city with dark, greasy soot. With the new, darker habitat, predators could easily spot the light-colored moths, and any moths born with the dark color mutation were much more likely to survive long enough to reproduce. Over time, the population shifted from nearly all light-colored moths to nearly all dark moths. Other examples of directional selection include Darwin's famous population of finches, who were more likely to survive if their beaks were better suited to the food that was most readily available. In stabilizing selection, an intermediate phenotype is favored rather than one or both extremes. A great example is a peacock tail. Male peacocks are famous for their enormous, colorful tails, which they use to attract female peacocks. Usually, females prefer the males with the largest and most highly colorful tails because they signal that the male is healthy and probably has good genes. Based on this, we might assume that the goal is to have as large of a tail as possible. However, in addition to attracting all the ladies, enormous tails have a couple of significant drawbacks. Males with exceptionally large tails are much more likely to be eaten before they have a chance to pass on their genes. The best of both worlds is to have a tail that is large enough to attract a mate, but not so large as to prevent you from flying. Male peacocks actually can fold up their tails and fly, but not very far. Other examples of stabilizing selection include birth weight in humans and stem height in plants. In both cases, the intermediate is favored. In diversifying selection, both extremes are selected for simultaneously, while the intermediate is selected against. Diversifying selection is much more rare relative to directional and stabilizing selection. If you think about it, it's pretty rare to have two extreme phenotypes that are equally useful at the same time, but it does happen. Rock pocket mice are an excellent example of diversifying selection. Much of the southwestern U.S. is made up of hot, dry desert climate with light-colored sand. The mice tend to have light-colored coats, and it allows them to hide from predators. However, in an area called the Valley of Fires in New Mexico, the light sand is suddenly punctuated by a large area of dark rock. This area is known as the El Malpais lava flow. Scientists believe that it is from a volcanic eruption that occurred roughly 5,000 years ago. In the area surrounding the lava flow, dark-colored rock pocket mice can also be found. Prior to the lava flow, any rock pocket mouse born in the desert with a dark coat as a result of random mutation likely would not survive long enough to reproduce. It would stand out against the light-colored sand and would be an easy target for predators. When the lava flow suddenly added areas of darker habitats, dark-colored mice suddenly were better able to survive. Today, across the Valley of the Fires region, we can now find both light and dark-colored mice. But what about intermediately colored mice? A mouse with an intermediate coat would not be well suited to either the light sandy environment or the dark rocky environment. Because it would struggle to hide from predators, it would likely not survive long enough to reproduce. Because both the light and dark extremes are favored, our final population graph would look like this.
Figure 1910 is just giving some examples of sexual dimorphism in peacocks and peahens. Species of spider, this one is, the large one is the female, and then ducks. So this one with the vibrant colors is the male, and this kind of brownish one is the female. Sexual selection refers to the pressures males and females face in obtaining mates. It can lead to the development of secondary sexual characteristics that don't necessarily improve an individual's chances of survival, but it does improve their reproductive success. Sometimes sexual selection is so intense that it favors traits that are actually detrimental to survival. Take the peacock's tail, for example. It's stunning, and the males with the largest, most colorful tail is most likely to attract a mate. However, it's not very practical, and it makes males more visible to predators and hampers their escape ability. There's evidence suggesting that females prefer big tails because they indicate a male's ability to survive with such a risk. The bigger the tail, the fitter the male. This is known as the handicap principle. The handicap principle suggests certain traits or behaviors in individuals can serve as honest signals of their genetic fitness or overall quality. According to this principle, individuals with more pronounced or extravagant traits, which may appear detrimental or costly, demonstrate their ability to survive and reproduce despite these drawbacks. These traits act as handicaps that put individuals at a disadvantage in survival, but they also demonstrate their genetic superiority and resilience. According to the good genes hypothesis, males develop impressive ornaments to showcase their efficient metabolism or their disease-fighting abilities. Females then choose males with these traits as it signals their genetic superiority, which would be passed on to their offspring. While some may argue that females shouldn't be picky to maximize their number of offspring, choosing better males can be beneficial. Having fewer but healthier offspring can increase their chances of survival more than having many weaker offspring. Both the handicap principle and the good genes hypothesis rely on traits being honest signals of male quality. This allows females to select the fittest mates who will pass on the best genes to their offspring. Figure 19.9 is showing that male yellow-throated side-botched lizard we were talking about before. He's smaller than the blue-throated or the orange-throated males, and it makes him look more like a female. And this allows him to be more inconspicuous with his mating choices. It's important to note that evolution doesn't have a predetermined goal. It's the accumulation of various forces influencing the population's genetic and phenotypic variation. Natural selection cannot produce a perfect organism. It can only make the best with what it has. It only works with existing genetic variation in a population and the new alleles that arise through mutation and gene flow. Natural selection can be constrained by relationships between different polymorphisms, and other forces like genetic drift and gene flow can introduce deleterious alleles to the gene pool, working against adaptation. Natural selection operates at the individual level, considering the net effect of alleles on an organism's fitness. Some alleles may be beneficial, others may be unfavorable, and their collective impact determines natural selection's influence. This means that good alleles can be lost if individuals carrying them also have overwhelmingly bad alleles. And bad alleles can be retained in a population if individuals with overall fitness benefits possess them. That concludes Lesson 10 material. Thank you guys for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.